Can somebody confirm you can hear me?
I can hear you.
Great. I've got another microphone error, so I'm not sure what's going on, but it seems like you guys can hear me, so the speaker is working. I think we're in chapter 8. No, maybe we're in chapter 13. Gosh, I didn't pick up where I left off. No, we did that. We talked about the operon model too. Hmm. Okay, are there any questions before we begin? Can somebody inform me, is this the last slide we talked about, or have I've talked beyond this, haven't I? Yeah, I remember talking about mutations. Plasmids. Have I talked about genes and evolution?
You touched on it briefly.
Okay. I think we are on chapter 13 then, although I gotta clean that one up, so let me leave it up.
I think we just started the new chapter.
All right. Uh, I believe I did talk about the consequences of lysogeny.
Yes, that's where I have left off.
Okay. All right. So we had gotten to here and said our next topic are animal viruses, viruses that infect animal cells. So let's talk a little bit about the multiplication of animal viruses. They follow the same basic pattern, bacterial phage multiplication. There are a few differences because obviously they're infecting different cells, animals instead of bacteria. The attachment, for example, happens to the cell membrane instead of the cell wall. Penetration is a little different too. It's much easier getting inside an animal cell because all it has is a cell membrane. So penetration of an animal virus happens by either endocytosis or fusion of the cell membrane to the lipid envelope of a virus. And we'll talk more about that. The uncoating can happen by viral or host enzymes. Biosynthesis is pretty much the same. You need to produce the nucleic acid of the virus and its proteins. Maturation is pretty much the same. Release is different, though, for animal viruses. In bacterial viruses, bacteriophages, we only had two life cycles, the lytic life cycle and the lysogenic life cycle. In animal viruses, we do have the lytic life cycle, but we also have the budding life cycle. And there's a few other life cycles we'll talk about. Persistent infection life cycle, just briefly, we'll talk about that. And then the latent life cycle, which the cells don't, or the virus doesn't really replicate in the latent life cycle. Let me see, most of the latent life cycle are budding viruses, but you don't need to know that. So the main two life cycles of animal viruses are the budding life cycle and the lytic life cycle. In the lytic life cycle, the virus will replicate and then it'll lyse the cell to release the viruses. So that is very similar to the lytic life cycle of bacteriophages. The budding life cycle is totally new. The budding life cycle only happens in enveloped viruses. And we'll explain why in a minute. Any questions about any of that? So the multiplication of animal viruses follows the same basic pattern, bacteriophage multiplication, but there are several differences. The main difference is that animal virus can replicate in either the nucleus or in the cytoplasm. Bacterial phages can only replicate in the cytoplasm because that's all there is to a bacteria cell. But animal viruses can replicate in the nucleus or they can replicate in the cytoplasm. And that's the main difference. Like I said, there's a few other differences like getting into the cell. Attachment happens, the virus attaches to the cell membrane, and the cell membrane will have the receptors for the virus to attach to. And the viruses will still have the ligands or the adhesion molecules on the virus, which will attach to the host cell receptor. But once the virus attaches to the cell, it will either enter by endocytosis or by fusion of the cell membrane to the viral envelope. Any question about any of that? Now, if anyone has purchased an older version of the textbook, and that would be the custom edition 1 or custom edition 2 for Clark College, the textbook has an error and it says that the virus enters by pinocytosis. That isn't correct. It enters by endocytosis or by fusion. Any question about any of that? All right, this is sort of showing you endocytosis, and that is the virus are these little round things attaching to the cell membrane right there. And once they attach, the cell membrane will invaginate surrounding the virus, and then this will pinch off. That'll make the vesicle surrounding the virus, and then that will pinch off and reform the cell membrane. And then this vesicle will move inside the cell. And that's how the cell, or the virus enters the cell by endocytosis. Any question about any of that? I will show you how the virus enters the cell by fusion of the viral envelope to the cell membrane. So there's the cell membrane. I'm going to make it a double membrane because that's what it is. Two lipid bilayers. I'm not going to draw it, but there will be a phosphate head here, the tail's coming to the inside, and then a phosphate head on the outside of the cell membrane, the tail's going to the inside. Any question about that? And then we have a virus right here that has a envelope, and it's a lipid bilayer. So there's the lipid bilayer. And then the protein coat will be inside of that. We'll make it something like that. And then the nucleic acid will be inside of that. What happens is, I should have drawn this a little closer. Let me expand this. is the cell membrane will touch the lipid bilayer of the envelope, and then that will pinch off or fuse. Let me see, how does that work? There we go. And so that lipid bilayer of the virus fuses with the cell membrane. Trying to draw that in there. There we go. And so this outer membrane now goes around the virus before it joins back to the cell. And then the inner membrane will fuse to that membrane. Kind of have it drawn a little tight there. And you can see that the cytoplasm now is connected to the virus capsid. And this can open up, and then the virus can move freely into the cytoplasm of the cell. Any questions about fusion of the viral envelope to the cell membrane? And why this can happen is because the cell membrane is a lipid bilayer mainly made up of phospholipids, and then the viral envelope is mainly a lipid bilayer mostly made up of phospholipids. Any questions about any of that? All right. And that's showing you the taking in of the virus into the cell. The vesicle has to be degraded to allow the virus to be released into the cell. So that's attachment, penetration, and coding is very much the same. It can happen by viral or host enzyme. Biosynthesis, very much the same as a bacterial virus. The virus will have to produce its nucleic acid for its genome, and it'll also have to make proteins. Like if this is a DNA genome, it'll have to be transcribed into messenger RNA, viral messenger RNA, by the way, which will then be transcribed into viral proteins. And then the viral proteins will have to get together with the viral genome, and that happens in maturation. The viral proteins self-assemble in the cell, and the virus makes an enzyme which actually stuffs the viral genome into the virus. All right. And then the lytic life cycle of a lytic life cycle animal virus follows pretty much the lytic life cycle of a bacteriophage, meaning the virus replicates and the virus destroys the cell, and then that releases the newly replicated virions, which then go out and infect other cells. So the lytic life cycle ruptures the cell, and this is done by non-enveloped viruses, meaning naked animal viruses. The budding life cycle is a little different, and it occurs with enveloped viruses. Any question about any of that? So the budding life cycle happens with enveloped viruses, and the lytic life cycle happens with naked animal viruses. In the budding life cycle, the virus will replicate, but at low numbers. It does not replicate at high numbers, at least most of the time. There could be some exceptions like in HIV, in the when the patient is in AIDS and the virus is multiplying like crazy, there might be a lot of viral multiplication. But normally in the budding life cycle, even in HIV, when it's not causing AIDS, it will be released by the budding life cycle at low numbers, so that when the virus buds out of the cell, it does not kill the cell. And we'll explain that in just a minute. So the virus will replicate at low numbers, and it will obviously replicate its nucleic acid, which you can't see inside of it, and the viral proteins, which is shown right there, sort of. And this is a complex virus because it's taking on an odd shape. And then the virus will come up to a membrane. This could be the cell membrane, it could be the nuclear membrane, or it could be the membrane of the endoplasmic reticulum. It just depends on what species of virus we're talking about. But a budding virus will come up to one of the animal cells' membranes. And then it'll start pushing on that membrane. And then it'll push further on the membrane, creating a bud. And then it'll rip off part of that membrane, which will reseal. So the cell membrane is not damaged, it is reseals. And then that cell membrane will reseal and form the envelope around the virus. And that is how envelope viruses get their envelope. It is literally a piece of a membrane, either the cell membrane, the nuclear membrane, or the membrane of the endoplasmic reticulum, which is, I don't know, budded off from the membrane to form the envelope of the virus. Any question about any of that? Let me show you this in a little bigger detail. This is a cell right there. And that's probably a cell, and that's probably a cell right there. In these cells, they are infected with the virus, and the virus is budding out of the cells. So there's a bud, 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 there's a bud. As I stated, the buds usually are not in large number, and when they bud out of the cell, they usually do not kill the cell that they bud out of. If the cell is killed, it usually is killed by the host immune system, recognizing that this cell is infected, and then the host immune system will try and kill that cell to clear the body of the viruses. Any question about any of that? And then there's one other thing I need to talk about, and that is, how does the host immune system recognize this cell is infected? Well, this cell will make viral proteins and glycoproteins, which will be put into the membrane. So if this is the cell membrane here, it'll have glycoproteins or proteins, viral proteins, put in it. And yes, the virus will actually move to a region of the cell that has the membrane of the cell that has the viral proteins and glycoproteins. So that those proteins and glycoproteins will then bud out of the cell and be glycoproteins and proteins in the envelope of the virus. And it's these glycoproteins and proteins that the host immune system usually recognizes as foreign, and then will mount an immune response against this cell. Any question about any of that? So these glycoproteins and proteins aren't found everywhere on the membrane, although they usually are found at lower numbers, like right here. It's not found at great numbers right there. And the virus will come up to a region where there's lots of these viral proteins and glycoproteins, and then bud out of this region. It won't bud out of, I forget my mouse to work. Come on, mouse. It won't bud out of this region here. It'll bud out of that region there. And then the virus envelope will have viral proteins and glycoproteins in it. Any question about any of that? Let me back up one moment. And these viral proteins and glycoproteins can make the spikes in the envelope viruses. All right, viruses have different genomes, either double-stranded DNA, single-stranded DNA, double-stranded RNA, or single-stranded RNA. Therefore, each virus type will need a slightly different mechanism for multiplication and biosynthesis. Double-strand DNA is pretty obvious how that replicates. The two strands are separated into single strands, and then each single strand is copied to make the corollary strand. Any question about any of that? All right, so let's talk about the DNA viruses. I should back up and state that there can be DNA viruses and RNA viruses. Let me blow this slide up. There we go. So there are 7 families of DNA viruses that cause infections and diseases in humans. Here are the family names. We won't talk about most of them. We will briefly talk about Poxviridae because it has smallpox in it. We will talk a little bit about herpes viridae. In fact, I've already talked a little bit about herpes viridae. Papillomaviridae, we won't talk about. Polyomaviridae. Adenoviridae, I'll talk just briefly about. It's one of the 3 families of viruses that cause the human cold. And the Adenoviridae is the least most common family that causes the human colds. We'll talk about the other 2 in a minute. And then the Hepaviridae I won't talk about, or the Paroviridae. You don't need to really know the DNA type, double or single-stranded. I don't need to know really whether the virus is enveloped or naked. I will mention herpes viridae and poxviridae are enveloped. And you should know smallpox is a member of the Poxviridae, pox family of viruses. And then the herpes simplex viruses are the member of the herpes viridae family of viruses. Any question about any of them? If you've heard of Epstein-Barr virus, cytomegalovirus, oh, what's the other one? Oh, there it is, roseola virus causing roseola. These are all members of the herpes viridae family. And there's a picture of a herpes simplex virus. There's a picture of adenovirus. Remember, adenovirus is the 3rd family or the least most common viruses that cause the human colds. For DNA viruses, the DNA is generally copied in the nucleus of an animal cell. However, the capsids and the proteins are synthesized in the cytoplasm using the host cell enzymes and ribosomes, as well as tRNAs. And this is a rule of thumb. A DNA animal virus replicates in the nucleus. And then the other rule of thumb is a RNA family of virus. We'll talk about it in a minute. The rule of thumb is it replicates in the cytoplasm, although there is one exception. for that, for the RNA viruses. Any question about that? Why do DNA viruses normally replicate in the nucleus? Well, in the nucleus of an animal cell, we have all the enzymes and machinery like ATP, the nucleotides, all of those are present for replicating DNA. And so animal viruses tend to replicate in the nucleus. And I don't know of any DNA animal virus that does not replicate in the nucleus. Now, there could be one that I don't know of, but I don't know of any. So it's a pretty good rule of thumb. I don't know of any exceptions, and they don't talk about any exceptions. Once the viral DNA enters the host nucleus, one of two things can happen. Let's talk about two first. The viral DNA can be expressed, producing no virions. That happens when the genome will be replicated and the genome will be transcribed to make viral messenger RNA, which will be translated to make viral proteins, which will need to get together with the viral genome to make the newly synthesized progeny virions. Any question about any of that? All right, so let's look at multiplication of a DNA virus in more detail. We have a DNA virus here, and it will bind to the cell membrane of a host cell by specifically having ligands or adhesions, molecules on the outermost regions of the virus. They will bind to the cell receptors on the cell membrane, which can be proteins or glycoproteins. It's just what the virus is binding to. The virus will then get in the cell. We're not going to talk about how that happens. It could happen by either fusion of the viral envelope or by endocytosis. And then the viral genome moves into the nucleus. So the virus has to be encoded and all that stuff. The viral genome will then be transcribed into viral messenger RNA. The viral messenger RNA will then move into the cytoplasm. The viral genome will then be replicated, which we are showing 3 here. Initially, there's just 1. And then the capsid proteins of the protein code, as well as any other parts of the virus, will be made in the cytoplasm. And then the virus genome will get together with the viral protein to make the newly made virion, which will then either leave the cell by budding, which is not shown here, or it can leave the cell by lysing the cell in the lytic life cycle. The new virions are released, which can then bind to another animal cell. Any question about any of that? All right, remember if the virus leaves by budding, it usually only makes a few copies of itself, which will then bud out of the cell, not damaging the cell membrane, whatever membrane the virus is budding out of. Any question about any of them? All right, if not, let's move to the RNA family of viruses. The RNA family of viruses is a larger family containing 15 membranes or families shown here. And the RNA viruses replicate in the cytoplasm as a rule of thumb. And that's because the machinery for replicating RNA is in the cytoplasm. There is one exception to this rule of thumb where an RNA family does not replicate in the nucleus, and that is with, let's see if I can find it, Retroviridae. That's the exception. A family of viruses which have an RNA genome, but they do not replicate in the cytoplasm. They replicate in the nucleus. And that's the only exception I know of, and the only one you really need to know. Any question about any of that? All right, let's talk a little bit about the different families. We won't talk about any of these families. We'll briefly talk about Orthomyxoviridae. This is the family of viruses that cause the human flu, so influenza. We won't talk about Filoviridae, although it's kind of interesting, or Rhabdoviridae or Paramyxoviridae. We will talk a little bit about Retroviridae. These are the retroviruses that when they infect a cell, they have a RNA genome, but the virus does not replicate in the cytoplasm. The virus will enter the nucleus and then replicate in the nucleus. Any question about any of that? We probably should talk about Paramyxoviridae because it's a family that has a large number of early childhood diseases, or I should just say common childhood diseases like mumps, rubella, which is the hard measles, and then what do you call that? Paramyxoviridae, which doesn't probably tell you much. It's the one that causes infects your lungs and then causes you to have not a bark, but a cough like a seal bark.
Would it be whooping cough?
Yeah, that's it. Thank you. I was trying to remember the name of that. I said we should talk about that, but we won't. Coronaviridae, we'll talk slightly about that. Coronaviridae, as you know, is the family of viruses that COVID-19 is a member of. However, this family was known long before COVID-19 came into existence. And this family is the family of viruses that is the second most common family that causes the human colds. So Coronaviridae, most of the viruses that I know of cause the human colds. But it is the second most common. It is not the most common family of viruses that causes the human cold. Flavoviridae and Togaviridae are very interesting, but I won't talk about them. Astroviridae. Hepiviridae is what causes hepatitis B, but we won't talk about it. Calciviridae, we won't talk about. Picornaviridae, we will talk just briefly about. It's a very diverse, very large family of viruses that cause very different diseases, such as poliovirus, rhinoviruses, meaning the most common viruses that cause the human colds, and then hepatitis A virus. And right there, you can already see polio, the human cold, hepatitis A, that these viruses cause very different diseases in different parts of the body as well. Rhinoviridae only in the upper respiratory tract. Rhino means nose, by the way. Hepatitis A causing liver hepatitis, and then poliovirus, which largely hits human nerve cells, killing them. Any question about any of that? You do not need to know the different families of viruses, this DNA type, envelope or naked, and the size of the virus. You might learn some of the names if I talked about it specifically, and then talked about the different diseases that that virus causes. Any question about any of that? Not hearing anything, so let's move on. These are some RNA-containing animal viruses, and you can see that the viruses take on many different shapes as well as sizes. So multiplication of RNA viruses essentially is similar to the multiplication of DNA viruses. There are some differences because RNA is their genome instead of DNA. And then I already mentioned, as a rule of thumb, the viral RNA replicates in the cytoplasm if it is an RNA virus. And there's only one exception that I know of, and you need to know this exception, and that is family Retroviridae. It causes retroviruses or HIV. Only one or two families of virus, by the way, cause HIV. But the multiplication of RNA is different because we're starting with RNA and not with DNA. When we're talking about RNA family of viruses, it's important to distinguish, is the single strand of RNA the positive strand, or is the single strand of RNA the negative strand? The positive strand of RNA, the genome, can be read by the ribosome, and it will code AUG for the start codon. The negative or antisense strand of RNA, a virus, is the corollary to the positive strand. So it will not have AUG. I'm trying to do this in my head, AUG. So that would be UAC for the negative strand. The negative strand RNA cannot be read by the ribosome. So it cannot be translated into viral protein, at least not directly. So for DNA, it's pretty easy, especially if it's two strands of DNA. You just separate the two strands, make messenger RNA out of it, and then translate that messenger RNA into protein by simply moving the messenger RNA into the cytoplasm if it isn't there. If it's an RNA family of virus, the RNA will already be in the cytoplasm. And then we get the viral replication as the viral genome gets copied and the protein coat gets copied. So that's how DNA family of viruses replicate, which is very similar to the way our cells replicate, by the way. But if the RNA virus is double-strand RNA, it does replicate similar. It's just that you have two strands of RNA. One strand will be used as a template to make the other strand. And then the positive strand of RNA can be read by the ribosome as messenger RNA to make the viral proteins, which I'll make an arrow down here, making viral protein. Any question about any of that? Now, if the viral genome is stranded, and it is the positive single strand, this viral genome can be read by the ribosome to make viral proteins. Any question about any of that? All right, how the single strand, positive strand RNA virus replicates is it will make a corollary RNA molecule, and this will be negative strand RNA. And then the negative strand RNA is used to make newly synthesized positive strand RNA. And that's how positive strand RNA replicates. It makes a corollary strand, single strand, so it'll be negative in this case because we're talking about positive single strand RNA virus. It makes the negative strand and then uses the negative strand as a template to make more of the positive strand. And just to reiterate, the positive single strand RNA can be read by the ribosomes to make viral proteins. Now, if the RNA family of viruses is a negative strand RNA, that means this strand of RNA cannot be read by the ribosome, so amino acids will not be made directly from the negative strand RNA. Instead, the negative strand RNA will copy itself to make a positive single strand RNA. And then to make more of the negative strands, you use the positive template to make the negative strand viruses. Any question about any of that? All right, let's move on. So it doesn't matter if we're talking about negative strand or positive strand, single strand RNA virus. Both of them have to replicate their genome, and both of them have to replicate their proteins, especially the capsid proteins, which will then get together with the newly replicated genome to make the new infectious viruses, what we're going to call progeny viruses. All right, any question about any of that? For the positive single strand RNA virus, we do need to replicate the nucleus. And that's positive strand. What it'll do is make a negative strand, and then use the negative strand as a template to make more positive strands. The positive strand RNA can be read by the polymerase or the capsid to replicate the positive strand RNA virus. Any question about any of that? Sorry, my voice is getting hoarse. So let's take a look at the positive sense single strand RNA virus multiplication in greater detail. The virus is right here, and there's the RNA genome. It will attach to the host cell and then either fuse the envelope of the virus with the cell membrane, or the virus will be brought into the cell by endocytosis. And I should mention that you probably know this from A&P if you've taken it, that the virus, when they bind to the host cell receptors, the host cell receptors tend to come together so that when the cell or the virus is brought into the cell and then released freely into the cell, the virus will have many adhesion molecules, but they're only playing an important role in the binding of the virus to the host cell. Any question about any of that? All right, so this is positive single-strand RNA virus. It will get into the cytoplasm. The virus will make a negative strand RNA virus, and then that negative strand will be used as a template to replicate more of the first virus, meaning the virus that first showed infection by an agent and then viral multiplication. So more of the viral genome is made. The viral genome, as well as the original virus, can be read by the ribosome, and that's because it's positive strands, single-strand RNA virus, and that will make more proteins. And then the cell will self-assemble the viruses and then release the virus either by budding or by the lytic life cycle, where the virus will lyse the cell, releasing the viruses. And that is how a positive single-strand RNA virus multiplies. Any question about any of that? All right, let's talk about antisense strand RNA virus and how it multiplies. Once again, we have a virus attached to the host cell membrane of this cell, and it'll attach on something that's called the ligand by some authors, and other authors call it adhesion molecule. It is the molecule on, in this case, the virus, that will bind to the human cell. And so if you want to recycle, you should actually put all your paper and thin cardboard in the recycling. All right, the negative antisense RNA is then released from the vesicle. And when it's in the cytoplasm, it can start transcribing RNA and then eventually carving the meat off of the spare ribs. Similar to a negative antisense RNA virus replicating. So the viral genome is in the cytoplasm, and it cannot directly replicate by itself. It has to make a corollary, which in this case would be a negative strand RNA virus, perhaps an adenovirus. Is that right? I don't know. And once the negative strand makes the positive strand can now be used to increase the metabolism in your tube. I'm saying your tube because this will be very similar to what you're doing in the Unknown Project. And the other thing you could do is, for a person, you just throw water at them. And what I'm going to try and say here is that if the negative antisense RNA strand is replicated, this negative antisense strand can be read by the ribosome to transcribe the cell. And then the viral proteins will get made from the positive strand RNA intermediate, which would be the dark ones here. It can make the capsid proteins as well as encode for the release factor, meaning it'll start digesting the cell wall, and then watermelon will come into the cell. Not watermelon, water will come into the cell, causing the membrane to expand. If it's a cell membrane, it'll just expand up to the point where it cannot expand any further, and then it'll burst, releasing the cytoplasm inside the cell to outside the cell, and that will kill the cell. Then we have maturation, where the protein coat get together with the newly replicated viral genome, which in this case is negative single-strand antisense RNA. And then after that, the virus will be released from the cell either by budding or by the lytic life cycle. And then the virus can go and infect other human cells. Any question about any of that? Come on. I lost control of my mouse. There we go. All right, let's talk about the family retrovirus, family Retroviridae. This includes the lentiviruses like HIV-1 and HIV-2. Both of these viruses cause AIDS. And then feline leukemia virus is another lentivirus that you may have heard of. And it causes the wasting away and, I guess, dementia in cats. I'm not sure what you call it in cats, but where the brain no longer functions well, if at all. A feline leukemia virus does not cause AIDS, but it is frequently called kitty AIDS because when the cat has feline leukemia virus, the leukemia will make the patient sick, and then the cells are excreting a substance which adds to the patient's nauseousness. And I think you have to consume it. I don't think you can breathe it in, but I'm not positive. Retroviruses are unique in that they contain the gene for the enzyme reverse transcriptase. Reverse transcriptase is a protein that helps the cell replicate. And here we have a retrovirus. There's the envelope here and the spikes there. And then right there is the capsid around the viral genome. And you don't need to know this, but the lentiviruses have two copies. Both of them are positive strand, single-strand RNA. Inside the virus, we have another unique feature, and that is the protein. So I'm drawing a blank on the name of that protein now. I just said it too a little while ago. Oh, RT, reverse transcriptase. So no, it's not that. What the heck is its name? Reverse transcriptase is its name. Reverse transcriptase is a enzyme that can take single-strand RNA lowercase ss, uppercase RNA, meaning for single-strand RNA. And this is a positive strand, so I guess I should put that in there too. Oop. Reverse transcriptase can take that and use it to make one strand of DNA. So reverse transcriptase will bind to this single strand, positive strand RNA virus of a lentivirus, meaning a virus like causes AIDS or something like that. And then start replicating the virus. And once the virus is replicated, it can use reverse transcriptase, which I guess I should put up there. RT for reverse transcriptase. To make DNA. Oh, wrong one. The textbook ignores this, but initially that's a single strand DNA molecule. Well, that needs to be over a little bit, but I'm going to leave it. And then reverse transcriptase. Let me see if I can copy that. Nope. I don't see it anymore. Well, that was not good. Ah, a single strand RNA. Reverse transcriptase. Just abbreviating RT. Can make single strand DNA. From RNA. And then the reverse transcriptase can work on single strand DNA. To make double strand DNA. As I stated, your textbook ignores this last part, or maybe it's that part that they ignored, that the step doesn't happen immediately. It takes 2 steps to make double strand DNA. Now, this virus can then transcribe the double strand DNA genome into viral messenger RNA. Making it a little shorter. And that'll be positive strand, by the way. I haven't stated that, but that'll be positive strand. And that can translate, has to move into the cytoplasm, but it can translate. Oh, let's go here. And that'll make capsid proteins. Oops. Which will then assemble the virus assembles and then we'll have our newly replicated virus leaving the cell. And how does the virus leave the cell? Anyone remember?
Was it budding?
Yes. So it'll bud out of the cell. And this will be a virus which is positive strand. I guess I should put that there. I'll put it right there. Positive strand, single strand, RNA genome. All right, any questions about reverse transcriptase? And what it does, it's very odd, and that is it can make DNA from RNA. Any questions about that? This is the one time where the law of biology, where DNA is always copied to RNA, which is copied to protein, doesn't follow the normal flow. Where here we have RNA making DNA, and then that DNA makes more RNA, which then makes the virus. Any question about any of that? Retroviruses comes from the name reverse transcriptase. You take RE from reverse and TR from transcriptase, and you have pretty much retro. Obviously, the O has been added. So this is an RNA family of virus that replicates in the nucleus. And you can see why, because it has a DNA intermediate. Oops. Let me blow this up. So we have our positive single-strand RNA genome, reverse transcriptase in two steps. First, it makes single-strand DNA, and then it makes double-strand DNA. And then transcription can occur from the double-strand DNA to make positive single-strand RNA, which can be translated in the capsid proteins, making the new progeny virions, which will then bud out of the cell. So the retrovirus life cycle, we have a virus, and it will attach to the host cell by its ligands, also called adhesion molecules, binding to the receptors of the cell membrane. And then it will be brought into the cell. I believe HIV will fuse with the cell membrane, bringing the virus into the cell. The viral RNA will then use reverse transcriptase to make single-strand viral DNA. And then the reverse transcriptase will convert the single-strand viral DNA to double-strand viral DNA. I didn't mention it before, but the retroviruses will then integrate into the host cell's chromosome. So that viral DNA moves into the nucleus and then inserts itself into the chromosome. This happens fairly random, meaning even the same person can have the retrovirus integrate at different locations in their genome, in different cells. Let me word it that way. Any question about any of that? This is not called a prophage when the virus is inside the chromosome of an animal cell, because a prophage stands for a bacteriovirus, meaning bacteriophage is where we get the name phage. So this is called a provirus, not a prophage, because it's an animal virus. In the case of animal viruses, if the virus genome integrates into the host cell's DNA, it will stay there and remain a part of that host cell's DNA for the lifetime of the cell. Any question about any of that? Now, in humans, we actually have, it's either 5 or 6 retroviruses that have gotten into their human germline, and we pass these retroviruses from parent to offspring. However, the retroviruses, all 5 or 6 of them, have mutated or gotten errors in them, and so these viruses are non-functional. But humans have, it's 5 or 6 retroviruses in the human genome. The mouse has a retrovirus which has gotten into the germline of a mouse cell, and this can be passed from mother to offspring or father to offspring. And this virus is active and can replicate in the mother, the father, and in the offspring. Any question about any of that? All right. So the DNA, viral DNA, which is in the host chromosome, can be transcribed to make viral RNA, which will be positive strand and can then go into the cytoplasm to be translated into viral proteins, making the capsid proteins of the virus. The capsid proteins of the virus will then get together with the viral single-strand positive genome of the virus, which would be the same thing as the viral messenger RNA, to make the new virions. They'll assemble, and then it'll bud out of the cell, usually keeping this cell alive. The virus nearly all the time does not kill this cell. What kills the cell is the host immune system recognizing that this cell is infected, and it'll then kill the cell. Any question about that? So the budding is at low enough numbers that the ripping off of the membrane does not deplete the membrane that would kill the cell. Any question about viral multiplication in Retroviridae, family Retroviridae? If not, let's talk about the coronaviruses. This family is Coronaviridae, which I'm sure is known to you because of COVID-19. But as I stated, coronaviruses have been known to humans for a long time, and they are actually the second most common cause of human colds. If you're curious, coronaviruses usually hit young children. It can hit adults, obviously, but Usually, a lot of the childhood colds are caused by coronaviruses. And you might recognize this picture. This is the picture I selected for my microbiology course long before COVID-19 ever came out. So I've been using this picture just because I thought it was a cool picture. And it's showing you COVID. Well, this is actually coronavirus, but it's the same picture as COVID-19. So coronaviruses are a single strand, positive strand RNA virus, and they're enveloped. Besides causing the second most common cause of the colds, they also cause SARS, MERS, and COVID-19. All three of these are related in that they cause severe acute respiratory syndrome. In fact, SARS stands for severe acute respiratory syndrome. MERS stands for Middle East Respiratory Syndrome. And COVID-19 actually stands for CO for coronavirus, ID for identification in 2019. That's what the 19 comes from. MERS has the highest mortality rate, where around 36% of the people who get MERS die. But fortunately, MERS does not spread from person to person. MERS mostly spreads from camels to people. So MERS is a virus that apparently grows in people, and then people who spend a lot of time around camels tend to come down with MERS. I mean, the patients who get MERS tend to be people who spend a lot of time around camels. SARS was a new virus that hit the world in 2003, and then it came back, meaning they got control of it. I think it spread to like 3 different continents. It started in China and spread fairly widely in China the first time, and then it spread to at least 3 other continents. North America was one of them. I'm not sure what the other 2 continents, obviously Asia, North America, and then one other continent. Unfortunately, they got control of it and then made it extinct in the human population. But later, I don't remember when, 2010, 2012, something like that, it came back and started spreading again in the human population. Fortunately, this time it only spread in China, and the Chinese were able to quarantine the disease and get it extinct and stop spreading in the human population. And this was a good thing because it had a mortality rate of about 10%. The good thing about MERS and SARS is everybody, or at least all the humans who get the disease or the virus, have signs or symptoms of the disease, meaning they have signs or symptoms of SARS or MERS. With COVID-19, the reason why it spread so readily is about 50% of young people, children, teenagers, and young adults, will get COVID-19 and they will be asymptomatic. And even though they will not have any severe signs or symptoms because they have no signs or symptoms, the COVID-19 can be growing in them and can spread from them. And they can take it to, let's say, their grandparents, which if they get COVID-19, it could be a very serious disease. The mortality rate of COVID-19 was originally something between 0.02% to 3%. But after, let me see if I can remember the name of that one. I'm not remembering the name of that one. After one of the variants of COVID-19 came out, it was the least lethal COVID-19. So the lethality rate is dropped by about half. So it's something like 0.1% of people who get it to up to 1.5% of people who get it can die from COVID-19. Omicron, that was the variant. When Omicron came around, the mortality rate of COVID-19 decreased. And the reason why Omicron was less lethal was because Omicron was actually a merger of COVID-19 and a coronavirus causing the human colds. And then these two viruses, I don't know, came together, making a new variant of COVID-19. And it's obviously got part of the genome from the coronavirus causing the human cold and part of the genome from COVID-19. And that's why that variant has been less lethal. Unfortunately, ever since Omicron has come about, all the variants of COVID-19 have been descendants of Omicron. COVID-19 spreads more easily than the flu, and one person is likely to infect at least 1 to 2.5 other people, which is why COVID-19 continues spreading. Actually, COVID-19 has, if anything, increased its easiness to spread. So it's probably higher than this now. Maybe it's spreading to infecting 1.5 people to something like 3.5 people, something like that. So this number is probably a little out of date. COVID-19 does have another name. Its scientific name is Severe Acute Respiratory Syndrome Coronavirus 2, or SARS-CoV 2. This name is so similar to SARS that even scientists do not tend to call it this. They tend to call it COVID-19. COVID-19 is closely related to bat coronaviruses and likely originated from two different bat coronaviruses that recombine, and then that recombined virus recombined with a pangolin coronavirus to make COVID-19. The pangolin portion of COVID-19 is very small, only about 10% of the genome, but it codes for the spikes, which is why COVID-19 spread so readily among humans. The spikes are what the COVID-19 attaches to our cells and then fuses to our cells so that the COVID-19 comes inside our cells. The pangolin is a spiny anteater, and a pangolin was likely an intermediate host between the bats and humans, meaning a pangolin probably was the first animal that infected the first person. And this happened in, I think it's Wuhan, I think it's Wuhan in China, which is the same place where SARS struck the world twice. And the reason why it happened in China is because the Chinese have these exotic animal markets where the markets are crowded with people buying products, and they're crowded with animals being sold in the animal markets. And having all of these animals and people close together probably increases the chance that a virus, which is spreading among animals, can jump species and infect humans. And the Chinese have realized that now 3 times they've had a lethal virus start in China from the animal market. SARS twice, and then COVID-19 once. And so they've actually passed a law and started shutting down the the weird animals in the animal markets in China. I mean, you can still probably get chickens and pigs and other domestic animals, but my guess is you would not be able to get a pangolin or any other weird exotic animal. Any question about any of that? All right, COVID-19 is transmitted primarily via droplets from coughs and sneezes within a range of about 6 feet. It may spread from indirect contact via contaminated surfaces. It's also in feces, although spread that way is not likely. But if somebody coughs on their hand and then touches a doorknob, it could spread that way. Fortunately, COVID-19 prefers to spread directly from the air by either colds or sneezes, or it can spread, not as likely, but it can spread from talking or especially singing. Springsteen does spread very easily from singing. So COVID-19 is likely breathed in. Rarely is it picked up by the hands and then self-inoculated, meaning you pick it up on your hands and then you inoculate a mucous membrane in your face. COVID-19, though, can survive for 3 days on plastic or metal surfaces, but only for a day on paper or cardboard. It does cause symptoms similar to the flu. Initially, runny noses and sneezing were less common. Actually, with the new variants of COVID-19, the runny nose and the sneezing are more common than it used to be. So it's very much like the flu now. Shortness of breath and deaths are more common than the flu. The mortality rate increases with age of the patient, ill health of the patient. Mortality is more common in men, and mortality is more common in Blacks and Hispanics. The last explanation for why COVID-19 is more mortal in Blacks and Hispanics has not been explained. However, it's been verified that the United States has seen it, Canada has seen it, and Mexico has seen it as well. The incubation period before the patient has any signs or symptoms of COVID-19, they have to get infected 2 to 14 days before that time. And then after 2 to 14 days, they have signs or symptoms. If they have signs or symptoms, as I mentioned, among young people, and that's most of you guys, about 50% of young people will be asymptomatic. And like I said, this actual death rate, that's what estimated worldwide. In the United States, it used to be 1.8%. It's probably about half of that now. Prevention, have correct hand washing, keep your distance from people, especially if they're coughing. Don't touch one's own face. If you're around people who may be sick, you should wear a mask. Not only you, but the person who's sick. The mask decreases your chance of getting infected, but it also greatly decreases the chance that the virus will come out of a sick person. It's best to stay home if you're sick, cough into your tissue or your elbow. Any questions about COVID-19? All right, if not, let's move on to viruses and cancer. Some human cancers are actually caused by viruses. And what we're talking about is where the patient gets a viral infection and then later the patient gets cancer. So a very strong correlation. Some examples: human papillomaviruses cause cervical cancer and skin cancer, and hepatitis B virus and hepatitis C virus are known to cause liver cancer. Any question about any of those? Not all cancers are caused by viruses. In a study by Tang in 2013, he found no cases of breast cancer or glioblastoma that were associated with a viral infection.
Do
But
you know off
some
the top of your head what kind of skin cancer the HPV causes? It's okay if you don't, I can Google it.
it usually causes a wart,
Oh,
and then
okay.
the wart grows and becomes cancer. I don't remember if it's a basal cell carcinoma or a squamous cell carcinoma, but it usually starts as a wart, and then the wart just converts into a cancer.
Gotcha. Okay, thank you.
I'm not going to talk much about cancer, but I will say that cancer is simply uncontrolled growth of your own cells. There are two main ways that viruses can cause cancer. The first way is a virus can form a provirus, where the viral genome actually integrates into the host cell's genome. This can cause a permanent genetic change in the cell, including causing mutation, turning on genes that are normally turned off, or turning off genes that are normally turned on, meaning you're causing a permanent genetic change in the cell, mucking up the DNA. And this is believed for why retroviruses have an increased risk of certain forms of cancer. The retrovirus integrates into the host cell's DNA at different locations in the different cells and just mucks up the DNA. Any question about any of that? All right, that's the first way a virus can cause cancer. The second way a virus can cause cancer is from an oncogenic virus. This is a virus carrying an oncogene. An oncogene is a mutated form of a normal cellular gene involved in cell growth. So we have a normal gene regulating and controlling the cells growing. And the mutated gene simply causes the cell to grow uncontrollably. A mutated oncogene can help transform a normal cell into a cancer cell. Because you have a normal cell, which can grow, obviously, our skin cell grows, for example. But it gets infected by an oncogene, that skin cell can then now grow uncontrollably. And that can lead to the formation of a cancer. There's obviously other steps. We're not going to talk about that. A cancer cell needs, it's like 6 or 7 mutations from a normal cell to change into a cancer cell. And the oncogene probably only brings in about 1 mutation. So the oncogene can transform the cell, so the cell can then grow uncontrollably. Further mutations can cause that cell lineage to transform into a cancer. And that oncogene gives the virus a benefit. You don't need to know this, but the oncogene causes the cell that the virus has infected to grow uncontrollably, which can help replicate the virus. And the virus then replicates in those cells. So those are the two ways a virus can cause cancer. Forming a provirus, where the virus integrates in the host cell chromosome, mucking up the DNA. And then by giving the cell an oncogene, a mutated gene that causes the cell to grow uncontrollably. Any question about any of that? All right, I mentioned that the two main ways that viruses can, excuse me here. the two main ways that viruses can replicate in a cell that we call a life cycle is by the budding life cycle or the lytic life cycle. But there are other life cycles. There is a latent viral infection where the virus, when it becomes latent, it leaves the budding life cycle, enters the latent life cycle. The virus remains asymptomatic in the host cell for long periods of time. When the virus is latent, it does not cause disease. The virus can then leave latency and enter the budding life cycle where the virus can cause disease as well as replicate. And we have two examples of that. When you have a cold sore that's caused by herpes simplex virus type 1, and while you have the cold sore, the virus is in the budding life cycle. The virus is actively replicating. And you're getting the virus to cause disease, the cold sore. Any question about any of that? The cold sore goes away when the virus enters the latent life cycle. The virus is just latent, staying inactive. It's still in the cell, and it can come out later. But while it's in the latent life cycle, the virus is not replicating, does not cause any signs or symptoms of disease. And then the other good example of a latent virus is the chickenpox virus. When a child first gets the chickenpox virus, they'll have the virus in the budding life cycle, replicating, causing disease, which will be the child's chickenpox, where the child is actively replicating viruses. And then the virus can go latent, and it can stay latent for decades. And usually the virus doesn't come out until the child is now an elderly adult. And when the virus leaves the latent life cycle and enters the budding life cycle, the virus will then replicate, causing signs and symptoms of disease. And this disease we call shingles, which is very different in the signs and symptoms of the chickenpox. But it is the same virus. Any question about any of that? There is usually a trigger for disease manifestation in a latent virus life cycle. With the trigger for the cold sores, it's usually the patient gets a cold, which then causes the virus to leave latency, enter the budding life cycle, and the patient gets a cold sore. And that's why it's called the cold sore, because you get these sores usually when you have a cold. There can be other triggers, though. In shingles, the trigger is usually the patient gets very elderly and their immune system weakens. And that's the trigger. Any question about that? All right. So that's the latent life cycle. There's also a persistent viral life cycle, and it follows sort of the budding life cycle, except, maybe I can draw it, except it's done by a naked virus. Why didn't that work? So we have a naked virus. And it will replicate in a cell, usually at low numbers. The virus will then assemble and it will be put in a vesicle. The vesicle then moves up to the cell membrane to release the virus. There it is. And there usually be more than one virus in a vesicle. So the virus is leaving the cell and not killing the cell, and is usually leaving the cell at low numbers compared to like a lytic virus. And I guess we'll just the release virus will then start the cycle again. We have a naked virus, which infects a cell, replicates in the cell, usually in low numbers, and will be put in a vesicle. That vesicle then takes the viruses up to the cell membrane. And so that's the persistent viral infection. These infections can last a long time, why it's called persistent viral infection. The disease of a persistent viral infection can occur over a long period of time. The detectable infectious virus may build up over long periods of time. Sometimes the virus is never cleared by the host immune system. Oop, let me get that there. So the virus starts at low numbers, builds up over time, and then it may bloom, causing disease. And you notice how this is broken? That's because it could kill the patient. HIV, before modern medicine, tended to be a virus that followed the persistent viral life cycle infection, meaning the virus would grow over time. It's actually a budding virus, by the way, not a persistent viral infection, but would follow the persistent viral infection and then would bloom when the patient had AIDS, producing lots of HIV, and then the AIDS would kill the patient while the graft is broken there. So the persistent viral infection may be fatal. HIV in the before modern medicine causing AIDS usually was fatal. With modern medicine, actually the HIV patient can live almost a normal lifespan. They can live something like maybe 5 or 7 years less than a normal individual, a healthy individual. But an HIV patient receiving modern medicine can live almost a normal lifespan. I'm sure you've never heard of subacute sclerosing panencephalitis, but if you have, it's actually caused by the measles virus, and it can, when it causes subacute sclerosing panencephalitis, be a persistent viral infection, getting worse and worse, and can be fatal. Many of the persistent viral infections can be tolerated, like cytomegalovirus, chickenpox, or varicella zoster. When my mother got shingles, she had it for 3 years for the rest of her life. And what happened was it was a persistent viral infection. and she always had the shingles. There are some other herpes viruses which cause a persistent viral infection, and they are tolerated, meaning they don't follow this curve. Instead, they follow a curve. Let me draw it out for you. They follow a curve like this, where over time, oops, it's not a very good graph. Over time, they slowly increase, and then they plateau out. It's trying to be a plateau there. Not a very good plateau, but they slowly increase, and then they plateau out so that they cause an infection persistently. Cytomegalovirus can do that. Anyways, there are different viral infections. There is an acute viral infection, which would be an example of the human cold or maybe even the flu, where the patient first gets infected, and then very shortly thereafter, they have signs or symptoms, which can be 2 or 3 days, up to 14 days. And then they have a very increase in the number of infective virions, meaning the viruses are being replicated at high numbers for a short period of time. And then the virus quickly decreases and then goes away. The human cold. So you have signs or symptoms of cold right here. And then it goes away. An acute infection. It differs from a persistent infection, which it tends to build up over time, over a long period of time, not just a few days, but months or years. And if it's tolerated, it'll just plateau out at some level. And if it is fatal, it'll usually bloom, causing serious disease, and then the disease will kill the patient. And that's 2 infections. In between that, we have a latent infection where the patient will first get the chickenpox, and that'll be an acute infection. And the virus doesn't ever go away like the human cold is thought to do. The virus hides out in the ganglia of the cells of the patient. And then 15% of the patients who got the chickenpox will then later in life, usually when they're an elderly adult, get a latent infection that we call the shingles. And in most patients, it will be acute, meaning you'll get an increase in the number of viruses, and then it'll decrease and go away, becoming latent again. Any question about that?
What would long COVID go under?
That's a good question. It's not really a viral infection, they think, that's causing COVID. They think the COVID-19 is either interfering with some normal process in the body, or it's causing the immune system to constantly be attacking the body. So normally COVID-19 would be an acute infection and then would most likely disappear, but the signs and symptoms would be long lasting.
That's what I'm dealing with right now, so I was just curious.
That's what they think. Now, obviously, I haven't studied that recently, so I'm probably at least a year out of date, but they don't think that long COVID patients are actively growing COVID-19 and then releasing COVID-19. But they do have signs of like the cough and other signs and symptoms. You could tell me what they are. I don't remember.
Well, for me, I am actually one of the small percentage of people that are having digestive issues along with the cough. I get winded very easily, and I just overall don't feel very well.
Yeah, I'm sorry to hear that. You're one of
Oh,
the
that's okay.
unlucky person. I can tell you that you don't want to get COVID-19 because the more times you get it, the higher the chance that you'll get long-term COVID.
See, I've only tested positive once and I'm up to date on my boosters.
Oh, unfortunate.
I know I have a small child though, so I have a walking health hazard.
Well, if the child's unlikely to get COVID-19. They may be asymptomatic though.
True. Okay, thank you. I was just curious, that's
Yeah, like I
all.
said, they don't think that's actually the virus causing the long-term. I mean, it is the virus that started it, but what's causing the long-term signs and symptoms? They think it's something else. That either the body has gotten damaged, like maybe your lungs got damaged from COVID-19, and so you're not breathing as well as you used to.
And that would make sense being super tired, just being able to nod off anywhere.
Yes, that too.
Okay.
All right. How are we doing time-wise? We got a little bit of time. Let me see if I'm supposed to finish this today. I am not, so I'm going to stop here. And then we'll talk about prions next time. Any questions before I go? Let me state there is no lab today. I forgot to show you this at the very start of today. There is no lab today. I'll log in at 7 and I'll answer any questions until 7:15. But after 7:15 or the last student question is ended, whatever is later, I will log off. Okay, so if you have any questions, I'll be in the lab from 7 to 7:15. There is no lab today. You should be working on your unknown project. Any questions about anything? All right, if not, I'll see you next time.
Have a good night.
You too.